In this segment, we're going to talk about pharmacodynamics, PD, the other half of the pharmacokinetic-pharmacodynamic duo. So dynamics is about change. This is the study of not the PK. The PK was about how the drug got in, distributed, and got out. But notice we never considered what the drug was actually doing. How did it change physiology? Dynamics is about change. So this is about how the drug actually changes the physiology of the patient who's taking the drug. So let's remember that when we give a drug, we have a protein. That's going to be a protein. That's a 3D structure of a protein. Hope you recognize it. This is our target. For some reason, this target is causing problems. Maybe there's too much of it, it's too active, something is wrong, and we would like typically to inhibit it. Sometimes we want to turn it on. Very often we want to turn it off. The drug that we give, D, often will find a pocket in this target and sit there. And that will change the function of the target. And presumably it will change the function in a way that helps the patient's disease state get better. So it might tone it down, it might activate it, but it will in some way change it. Now, remember, the drug, when we take it, gets distributed throughout the body. And it goes everywhere. But somewhere in the body is the actual target that it was meant to bind. And so pharmacodynamics is the study of binding of the target and then the downstream pathway of effects that will lead to a change in the physiology. So this is just one protein. It might interact with another protein. So there's this protein, and it's interacting with this protein, and they do something together, and that perhaps leads to some other effect in the body, which in general will lead to a pathway of consequences of changing the function of, so this is A, this is B, and you might have C, D, etc. So these pathways of, I'm calling them consequences, these are physiological consequences. And pharmacodynamics is the study of how the drug works. In the end, these pathways will lead, in general, to a couple of things. They will lead to the therapeutic effect that we want. So it's a therapeutic effect. It may also lead to side effects, other effects that we may or may not want, usually don't want them. And the worst kinds of side effects may be adverse effects, where there's actually harm being done to the patient. All of these effects are a result of the drug binding its target, or sometimes binding what we call off-targets. So I was talking about how this drug was designed and tested to interfere with A. A was involved with the disease, and so by modulating A, you're helping the disease. But the other possibility is that there's another protein. Let's call it B. Looks different from A, but it also has a pocket, and drug D might also bind. I don't want to call it B because I invoked B before. So let's call this off-target, let's call it X. X might interact with Y and Z and have another set of physiological consequences. And these consequences may be not helping the patient. They may be side effects, or they may be adverse effects. The reason I draw these in parallel, in fact, let's make them look parallel, is because there's a big difference between side effects and adverse effects that result from the drug working with its intended target on its intended pathway. These would be called on-target therapeutic effects, on-target side effects, and on-target adverse effects. The unanticipated binding of the drug with an entirely different protein that's perhaps not part of that on-target pathway is going to be called an off-target pathway. This was not the target that was intended, and it's off-target. Therefore, the side effects and adverse effects, the way that they happen, is unrelated to the way that the drug is helping in the therapeutic setting. This is important in pharmacology because these off-target effects could potentially be engineered out by changing the drug so that it binds the target but no longer binds the off-target. So you can maybe work with these side effects and adverse effects to get rid of them or to lower them. It's going to be much harder to get rid of side effects and adverse effects that are part of how the drug works, because you really did want to bind this target and you really did want to have all of these effects. The problem is some of the good things that are happening are being counterbalanced by some of these side and adverse effects. They're going to be much harder to engineer out because it's kind of the cost of doing business with this pathway. So that's an important concept for pharmacodynamics. The other important concept in the setting of pharmacogenetics is that, again, all of these proteins in both on and off-target pathways are coded for by genes. And so you could imagine that if there are genetic variations in any of these proteins, it could change the way the drug binds. Let's just be real specific. I could change an amino acid in this binding pocket right there that changes how tightly D binds to its target A, and that could change the effectiveness of the drug. It could even change the balance between therapeutic effects and side effects or adverse effects. Similarly, there might be a mutation in B. There's 2 mutations in B that affect its ability to interact with A. That could change the response of the physiological system to the binding of D to A. And then, of course, the same arguments for off-targets. So in pharmacodynamics, we're studying how the drug binds its targets, how it then creates a pathway of effects leading to clinically measurable therapeutic effects, side effects, adverse effects. We've recognized that there are on-target and off-target effects, and it's this study that's important for understanding drug response and is important for understanding why genetic variation in any of these targets or the pathways can lead to a change in the experience of a drug.